Okay, so picture a hacker, but this hacker isn't human, and it doesn't use a computer. Today, we're diving into the incredible story of a tiny bacterium that's been genetically engineering plants for millions of years. And even better, we're going to talk about how scientists turned this natural foe into one of biotechnology's absolute best friends. It's a story that starts with a pretty weird and mysterious sight. It all begins right here. You've probably seen something like this on a tree or a shrub. This bizarre, lumpy growth called a crown gall. For centuries, people saw these plant tumors, but nobody had a clue what was causing them. I mean, it looks like something straight out of a sci-fi movie, right? But believe me, the reality is way more fascinating.
See, this isn't just some random disease. This is the handiwork of a microscopic, natural-born hacker. A true master of genetic code that figured out how to breach a plant's defenses and rewrite its very DNA for its own benefit. Yeah, this is a story of biological espionage.
So, let's meet our culprit. The organism responsible for these crown galls is a really common soil bacterium called Agrobacterium tumefaciens. It's practically everywhere, just living quietly in the soil, waiting for its chance.
And here's the key. Agrobacterium doesn't just make a plant sick, it physically transfers a piece of its own DNA into the plant's genome. It literally injects its genetic code into the plant's chromosomes, altering it permanently. This is what makes it a true, natural-born genetic engineer.
Now, figuring all this out was a massive biological detective story. Scientists knew something really strange was happening, but it took decades to piece together all the clues and understand exactly how this little bacterium was pulling off such an incredible trick. And this investigation was a total marathon, unfolding over more than 40 years. It kicked off in the 40s with a pretty simple observation. Whatever caused the tumor was creating a permanent change in the plant. They just called it the tumor-inducing principle. Then, by the 70s, they'd found the source, a circular piece of DNA inside the bacterium called the T plasmid. A few years later, boom, they narrowed it down even more, identifying the exact chunk of DNA being transferred, which they named tDNA. And the final piece of the puzzle fell into place in the 80s with the discovery of the virulence, the molecular machinery that basically acts like a syringe to inject that tDNA. So this is it, the T plasmid. You should think of this as the bacterium's own personal USB drive, and it's loaded with all the hacking software it'll ever need. Everything required to completely hijack the plant's cellular machinery is encoded right here on this little circle of DNA.
So let's break down this toolkit. First up, you've got the tDNA. That's the payload, the malicious code that actually gets injected. It's got oncogenes, which are what tell the plant cells to just divide like crazy and create that tumor. But it also has these opine synthesis genes. This is a super clever trick that forces the plant to produce a special food, called opines, that only Agrobacterium can eat. And finally, you have the virulence genes, or vir genes. Now, these don't get transferred themselves. They're the essential machinery that cuts, packages, and delivers the tDNA.
So how does this hack actually play out? Well, it's this beautifully efficient four-step process. First, the bacterium senses chemical signals from a wounded plant. That's its open door. This signal activates the vir genes. Second, the machinery makes a copy of the tDNA and wraps it up in protective proteins. Third, it uses a structure that works just like a tiny syringe to inject this DNA package right into the plant cell. And finally, that package makes its way to the nucleus and just slips itself into the plant's own DNA. And just like that, the hack is complete.
And this right here, this brings us to the single most important discovery in this entire story. Scientists realized that the viral machinery didn't actually care what was inside the tDNA. Nope, it only recognized the border sequences on either end. And that meant they could cut out the tumor-causing genes and replace them with, well, with anything they wanted. This was the aha moment that changed everything. Scientists could now hijack the hijacker. They could take this amazing natural genetic delivery system, which had evolved over millions of years, and reprogram it to deliver beneficial genes instead of the harmful ones.
And this is the core of that whole transformation. The original, wild plasmid, that's the foe. It's got oncogenes, it causes tumors, and it forces the plant to make its food. But scientists created an engineered version, the friend. They disarmed it by just taking out those oncogenes. So now, instead of causing a disease, it can carry any useful gene we choose, turning it into this fully customizable, incredible tool for genetic improvement.
So how did this all translate to the real world? Well, once this mechanism was harnessed, it completely revolutionized plant science and agriculture. It just opened up possibilities that were once pure science fiction.
And this isn't just theory, okay? This tech is behind some of the most famous genetically modified crops out there. The flavor sauvor tomato, that was designed for a longer shelf life. Bollgard cotton produces its own insecticide thanks to a gene from another bacterium. And of course, Roundup Ready crops that can withstand herbicides. All of these were made possible by using Agrobacterium as the delivery vehicle.
But its impact goes way beyond just making better crops. Agrobacterium has become a fundamental tool for basic research. Scientists use it for something called insertional mutagenesis, which is basically just letting the tDNA insert randomly into a plant's genome to break a gene. By seeing what goes wrong, they can figure out what that gene was supposed to do. It's an incredibly powerful way to discover what genes do and how they're switched on and off.
So when you trace it all the way back, it's just an amazing journey. We started with a weird, unexplained plant tumor. Through decades of brilliant scientific detective work, we uncovered a natural genetic hacker. And by understanding its methods, we transformed that plant foe into an indispensable friend, a true cornerstone of modern biotech.
It's really an incredible example of learning from nature to build groundbreaking technology. This perfectly designed system for inserting DNA wasn't invented in a lab. It was discovered hiding in the soil. Now the power to use it is in our hands, which leaves us with a pretty big question. Nature gave us the tool. What should we build with it next?